A very good afternoon, everyone. I, Dr. Dattathrayam Shinkar, Associate Professor, Department of Pharmaceutics. Good morning, Dr. Vaibhavkumar, sir. Now it's around 3 a.m. at USA. Welcome, Dr. Vaibhavkumar Gavali. A special thanks on behalf of our entire fraternity of GS, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, to Dr. Vaibhavkumar Gavali for his acceptance to share his expertise to all the participants. It is an honor to welcome you all on the occasion of this 1 week e-FDP and STP today. We have an extremely insightful session 2 of day 1 on topic electrophysiological methods for drug screening that promises to be both informative and inspiring. I feel honored to introduce speaker of session 2 of day 1, Dr. Vaibhav Kumar Gowri sir. He is a pharmacologist, cellular electrophysiologist and transnational research, Frontage Laboratory, USA. Sir completed his Doctor of Philosophy in Molecular Pharmacology from Medical University of Vienna, Austria in 2016, Master of Pharmacy, Pharmacology, Toxicology and Therapeutics from University of Mumbai, India in 2009, Bachelor of Pharmacy from SSDG College of Pharmacy, Chandwad, University of Pune, India in 2006. Sir is a recipient of Best Alumni Award in 2019, Nature Masterclass Scientific Writing and Publishing, Nature Publishing Group in 2021. He is also having the professional skill of computational statistics focusing on R from University of Cincinnati, Ohio in 2019. He is having vast experience. Now he is a lead scientist and study director, electrophysiology and safety pharmacology, Frontage Laboratory, Cleveland, Ohio, USA, from 2023 to present. He is a research scientist, one study director, electrophysiology and safety pharmacology, Charles River Laboratories, Cleveland, USA, from 2021 to 2023. He also served as a research scientist, internal medicine and neurology from August 2021 to October 2021 and postdoctoral fellow, internal medicine from January 2017 to July 2021 at College of Medicine, University of Cincinnati, Ohio, USA from 2017 to 2021. He is a PhD student and research staff at Medical University of Vienna, Austria from 2011 to 2016. Also serve as a trainee, research associate and junior research associate, Discovery Biology Team, Jubilant Biosys Limited, Bangalore, India, in year 2009 to 2011. Executive and trainee executive, Department of Medical Affairs, IPCA Laboratories, Mumbai, from 2008 to 2009. With respect to his technical skills, now manual and automated patch clamp electrophysiology, cytopatch, cytocentrics, and port a patch nanion. He also having skill with respect to live microscopy for calcium 2 plus assay and flow cytometry for membrane proteins expression in T lymphocytes. Maintenance of cell lines and primary and stem cell derived cells. Also having a vast knowledge of comprehensive in vitro pro-arrhythmia assay and FDA's ICH 7B guidelines. Worked on cell biology, transfection, siRNA, biophysical analysis of mutations, luciferase assays for cell signaling. He is also proficient in ClamFit, Graph, Prism, Sigma Plot, and Origin software. He is an incipient learner of R program also. Now, professional skills. He worked as a project management and leadership skills, experience managing multiple projects in industry as well as in academia. Innovation, drive innovation in projects, bring new ideas for grant writings. And with respect to science communication, he is also having a vast experience in guest editing of peer review journal and annual magazines, scientific writing for wider audience, public speaking on science and global research opportunities. Publications. Sir authored and contributed more than 24 peer review papers and 35 conference abstracts with more than 390 citations, He is also having Google Scholar H index of 12 and ITIN index of 16 with 395 citations to his credits. Now, I request Sir to share his views on topic electrophysiological methods for drug screening. With this brief introduction, I request Dr. Vaibhavkumar sir to kindly start your session. Please, sir.
Thank you. Thank you so much. Can you see the presentation slides?
Yes. Yeah, yeah.
Okay.
Visible and you are audible also, sir.
Okay, great. So I'm about to start. Technically, everything okay? Technically, everything okay? Can slide scrolling is visible? Just want some feedback.
Yes. Slides are
I
visible and scrolling also, sir. No issue.
Okay, great. Thank you. So I'll start and let me know if there's an interruption or any technical issue. Okay. Sir, just confirming. Hello. Mic test 1 2 3. Can you hear? Can you hear me?
Yes, sir.
Okay. Ready. So good afternoon, everyone, and thank you so much for joining. Um, Thank you, Dr. Shankar sir, for this generous and kind introduction about me. And first of all, I would like to join thanks to the management or the organizers from MS Gosavi College of Pharmacy. Also, thank you to Dr. Saibra Borasti sir for availing this opportunity for me to present the topic today and giving these insights on this unique field. Today, I'm going to speak about a special topic from cardiac safety pharmacology. It's about electrophysiological methods for drug screening. And in this presentation, I will be talking about the electrophysiology method itself, a brief introduction about what is electrophysiology, and the technical aspects of electrophysiology, including what this technique is about, where it is used, the details about the instrument and the procedure for drug testing and drug screening, and further regulatory aspects of this technique will be also be discussed. I will also share few examples about electrophysiology so that you are aware where this techniques is used, and related to pharmacology and applications of this technique in drug discovery and development process, and also its importance in terms of regulatory aspects, because I realize the background and the theme of this workshop or seminar is also focused on pharmacy education and regulatory perspective. So I would like to bring those elements to the presentation as well. And feel free to interrupt when you have a question I would like to answer during, or also we will have a question answer session at the end of the presentation. So going forward, so why electrophysiology? So there are some unique reasons why we do electrophysiology, and one of the reason is it's pretty fast, ultra fast. It has a microsecond resolution. It is highly accurate, and it's very versatile, and it's very interesting and sometimes very fun. So I have a small, cartoon of whole cell patch film electrophysiology, one of the confirmation configuration in the technique where a cell is shown and a tiny electrode touching the cell is shown. So that's how delicate this technique is. As you can see, the glass electrode is touching the cell membrane, and it has filled with the solution. So let's find out what this technique is about. Some of you may have heard about it, but there are more details to it, and we will see one at a time. So what we do in electrophysiology, basically, we measure two key events across the cell, or rather cell membrane. As you can see, um, a cell is shown, it has a charge, the negative inside, the positive inside on the membrane, and this inside diagram shows the view of the cell membrane. And this is the circuit, the electrical circuit, which shows the membrane acting as a capacitor, then there is a resistor, and then there are the electrical terminals. The VM represents the membrane potential difference. So in electrophysiology, we measure two things, or rather two events. One is the measure voltage across the biological membrane, so the potential difference. And the second one we measure is the current across the biological membrane. And the first experiment was conducted in 1950s by two British physiologists, Hodgkins and Huxley, and they also recorded nerve action potential during that time. But this technique evolved eventually, refined, and in 1991, two physiologists from Germany, Dr. Sackman and Neher from Germany, also won a Nobel Prize in Medicine or Physiology for their discovery of measurement of single channel currents using cell membrane, using this technique. So it's very special, and it has a broader application throughout the process of drug development, discovery, and also safety pharmacology and biophysics. So I'm going to talk about the two major aspects of this technique. One is current clamp, and one is voltage clamp. And in this technique, this is the in vitro technique at the moment I'm focusing is, so in vitro electrophysiology. And there's also in vivo electrophysiology, but the focus of this presentation will be on in vitro electrophysiology. And in short, this technique is called patch clamp. Instead of calling it patch clamp electrophysiology, I will be referring it as a patch clamp technique. So the patch clamp has two major configurations. The first one is current clamp. As you can see, a current is injected, a constant current is injected while measuring the membrane voltage. And this current clamp configuration is used to de or hyperpolarize cells. And basically, it measures the reaction of the cell in terms of membrane voltage. And the best example of current clamp is the action potential. So you can see in this diagram, we have a current step, and in response to current, the voltage is measured. So, and on this bottom diagram, you have this unique action potential cartoon shown, and there is this biophysical circuit shown. So current clamp is basically used for measuring the action potentials. And the practical example of the current clamp is in physiological scenario, the excitability of DRG neurons, so the dorsal root ganglion neurons. And this is a screenshot from a publication, Yusuf et al., where the authors have recorded the action potential of from a dorsal root ganglion neuron. As you can see in control situation, we have they have stimulated the neuron, and a series of current pulses were applied ranging from 0.1 nanoampere to 0.5 nanoamperes, increasing order. And there was a response of current to this injection, but it was smaller amplitude in case of control situation. But when they added the agonist, the stimulate the neuron, the ATP at 10 micromolar, and there was a rapid escalation of action potentials, so quite fast, which is very known in neurophysiology. So that was captured using current clap technique. So it's called a rapid burst of action potentials, or a rapid firing of action potentials in DRG neurons. And this technique allows us to record this phenomena. And once they washed out the ATP, the action potentials return to its normal state. So this is a very good example for current clamp. Then going forward, there are also two electrode voltage clamp that measures membrane voltage compared with desired voltage, and they inject current into the cell to bring the membrane potential. Represents this diagram shows there is a voltage electrode, there is a current electrode, and this is the electrical circuit representing two electrodes. So it's like injecting the currents using two electrodes. And here are some examples. So these are cells from frog oocytes. They're also called as xenopus oocytes, where um, I'm not sure the resolution if it is visible to you, but this cell, there is one electrode on the left side and one electrode on the right side. So this is a image taken from a microscope, so it may not be that clear, but you can imagine here. And this technique has some advantages. It is a fast clamp procedure, it is accurate, and it is very simple to perform. But there are some disadvantages, that it only works on larger cells like Xenopus laevis oocytes, these frog oocytes. And mammalian cells are sometimes smaller than this, so this is why 2 electrode voltage doesn't work with them. And that's why a further sophisticated technique is required, and it's established. It's called patch clamp. It has multiple configurations, and I'll talk about it one by one. First, let's see what exactly a patch clamp is. So this blue dot represents a cell, and this gray line is the electrode. So touching the cell using an electrode, and in this magnification you can see how different configurations work for this particular technique. So once the cell is touched using the electrode, this configuration is known as a cell attach mode, because it's simply touching the cell. And in this magnified view, you can see that the pipette is touching the cell. This blue line is cell membrane, and the gray cell is pipette here. The resistance is higher than 1 gigaohm, so it's very tight seal here. And from this point, again, it will go into two directions. The first direction is pull back. So once you touch the cell, then you gently pull the pipette with this pressure, and then the small piece of cell membrane will be disconnected from this whole cell, and there will be an exposure to only a brief part of cell membrane. So like few channels will be removed from the cell membrane, and we can measure the current from those tiny channels. And this technique allows us to record the activity of ions and across this single channel. So it's called inside out technique. And then another configuration from cell attach mode is, um, instead of pulling the pipette, keep the pipette there, and then just rupture the membrane by using a gentle suction here. And you can see it will open the cell membrane, and this technique will go into the whole cell membrane, whole cell mode. Then the pipette is pulled back after rupturing the cell. Its pieces of ion channel will also be pulled back, and it will be outside out. So outside of the cell will be exposed to the solution, and then we can apply the drug to these channels. So basically, inside out and outside out procedure. But most of the experiments in pharmacology are done in whole cell mode as well, because the whole cell mode will allow access to entire cell. As you can see in the this little picture shown in this slide, a cell is shown in the middle. It appears to be a neuronal cell where the dendrite procedures are seen, and there is a glass pipette, including the electrode, is touching the cell. And once the pipette touches, the cell membrane is ruptured, and then the whole membrane has electrical access to the pipette. Going forward, the advantages and disadvantages of patch clamp methods. The advantages are only one electrode is needed. Also, small cells can be studied. It has a very low noise, so less than a 1 picoampere. It's very versatile. It has different modes as explained. Disadvantages. Um, it is very sophisticated. Sometimes you need very fancy equipment. Stable and highly accurate micro manipulators are needed. Vibration isolation is needed. I'll talk about it in the next few slides why these techniques are required. And then accuracy of voltage clamp low for large currents. So when you have cells that have large currents and few nanoampere level, then the accuracy of voltage clamp decreases. So, but still, these patch clamps remain a gold standard for most of the pharmacology and biophysical experiments, and it is quite used regularly in drug development studies. So I wanted to walk you through the technical aspects itself, and wanted to give you or you wanted to visualize you how this equipment looks like and how the setup looks like, what things are needed, so that you will have an idea about how it works, and what are the things are required. So this is one of the setup I have used in my postdoctoral training, and I have obtained the permission to show you these figures here. So this is a big table, and this table is actually a floating table, because on the bottom itself there is a air insertion, so this table floats on the air. It's called floating table or anti-vibration table, and it is shielded by a special wire mesh to prevent the electrical noise um, during this sophisticated requirement. So this cage is called Faraday cage. And then there is a side view where we have a inverted microscope. As you can see, um, the objectives are under the stage and the lenses are here. And this little head stage here is shown where we fit the electrode, and this is the chamber, that's where we put the cover slip with cells on it. And there are some tubes that are used to transport or exchange the solutions, to perfuse the drugs, test solutions, and buffers. And here is the front view. As you can see, the recording chamber is clearly seen. There is a microscope light as well, and electrode immersed into the solution and touching the cell. And there is also a vacuum outlet, because when you perfuse the solution, there is a quite accumulation of fluids there, so you want to remove that thing. So that's why a vacuum outlet is also present. This is a very traditional gravity perfusion, which can be used in some other experiments like Langendorf and normal perfusion. So we can customize it based on our needs. Then we also need puller to pull the electrode or fabricate those tiny electrodes using a glass capillary. So the Sutter instrument is one of the company which uses this puller to fabricate. And this is a programmable puller, so The pipettes can be customized based on the experimental needs, especially the tip of the pipette, which is a unique feature of the experiment. It can be fabricated according to specific dimensions. So this is available in that configuration. And I would like to show you, so this was about the setup. I hope this brings some clarity on the technical aspects of the patch clamp electrophysiology, and it gives you some sort of idea that okay, how the setup looks, how the lab looks, what are the key things that requires in order to perform these patch clamp experiments. So, and we have already talked about the theoretical principles behind this technique. And let's talk about some applications to of patch clamp to screen the drugs, to develop the assays. So I'm going to show you some example. So practical examples, what can be done. So we have different types of ion channels in the cell membrane, and in there are mainly voltage gated ion channels and ligand gated ion channels. So patch clamp is one of the technique which can which allows us to record the activity of ion transport and drug action on these channels. And in pharmacology, you have different agonists and antagonists that can be characterized using this technique. So I'm going to show you some examples that are quite routinely used, you know, to characterize the drugs using patch clamp assay. So this is one of the example for ligand-gated ion channels. So acetylcholine, which is an agonist for muscarinic receptors, and these are the ligand-gated ion channel, and you can see dose-dependent effect of acetylcholine. So these are the currents measured using patch clamp, and you can see upon increasing concentration of acetylcholine, the current amplitude is stimulated from 10 micromolar to 3000 micromolar. We can see in effect on the muscarinic currents here. And on the bottom side, you can see a standard dose response to calculate the EC50 value for acetylcholine on this ligand gated channels. And there is also spontaneous events being recorded. Um, as we spoke about spontaneous events like single channel activity and also action potentials. So here in the control situation, you have these spikes. The spikes events represents the activity of channels. That means they were opening at these events. And when you add a antagonist or the blocker of the channel, these channels were blocked. So the spike events or the opening of the channels were also blocked, and they are no longer visible in this once you treat them with antagonist called hexamethymium. So this is one of the example. Then from ligand gated ion channels, I'm going to switch a voltage gated ion channel, where instead of ligand we will add voltage as a stimulant. As you can see in voltage gated ion channels, we have voltage as a stimulant. So I'm going to show you an interesting schematic representation how voltage gated ion channel respond to voltage and opening happens. So um, here in this figure, um, on the top here we have this pulse protocol where I'm going to hold this is the example where the cell was held at negative 90 millivolts, which is close to our body's resting membrane potential, and then a brief voltage pulse was given around negative 60 millivolts, so basically depolarizing the cell, changing the potential of the cell. And this is the current in response to that voltage. And in this graphical presentation, we have shown here on the y-axis, we have the current, and on the x-axis, we have the voltage as a stimulant. And we can see how it responds. So um, basically at negative 70, the channels are closed at resting state. That's why there is only flat line indicating the activity of channels were closed. But when you have further steps going to -60, there was a brief opening. When you further go around -40, the channel starts to open. The current starts to flow inside. This is the example of sodium channel, which are the typical voltage gated sodium channels. And you can see as we increase the test potential from -70 towards more depolarized potential, the channel starts to open, or in other words, it starts to activate. And you can see it's already go beyond half point of its activation, and at plus 10 millivolt is fully activated. So we already got the peak current here. It is also shown on this graph. And after that, there is a reverse flow of the current because the charges inside the cells are full and the current changes its direction. So it started going from inward to outward, and the net to maintain the net electrochemical balance in the cell. And you can see as we go further and further, the channels the current goes in the reverse direction. So I hope this example gave you an idea how voltage gated channels are characterized using this protocol in patch clamp. Then similar situation with voltage gated calcium channels. There is a small difference in the kinetics, but the principles remains the same. This is just some other snapshots from the published literature to show you that upon brief protocol, the channels are open. You can characterize different effects, different concentration of ions. In these examples, they have shown calcium versus barium to showcase that this channel was conductive to both of these elements. And then they have also used this equation to showcase the current voltage relationship. Some other example just to give you an idea that how we used patch clamp to characterize different types of membrane proteins. So we have seen already the voltage gated channel, ligand gated. Now further example is the G protein regulation, the GPCRs. So GPCRs like P2Y receptors, as you can see in the schematics, upon ligand binding like ATP, the channel opens, and we have increase in the channel activity, which is seen in this untreated scenario. And once you have the antagonist like a blocker or antagonist, here in this case the GPCR antagonist was used like pertussis toxin at a very very small concentration of 100 nanogram per ml, the currents were blocked. Going forward, same thing, GPCR with another example here. Here we are showing the effect of agonists like bradykinin. There was an increase in current upon increasing concentration and exposure to bradykinin. And there is also effect of antagonist also. Excuse me. Going forward, I would like to just give you a brief shot that this technique is also used in a diverse array of settings. So the major channels are voltage gated ion channels, but there are other minority groups of channels involved in a different cell types. And this technique is also used to study these channels and characterize their effects, characterize the effect of drugs. And this is also one of the example where activation of calcium dependent potassium channels, and these channels are present sometimes in excitable cells like muscle, heart and neuron, but sometimes it is also present in non-excitable cells like T lymphocytes. So this is one of the example of a moving T cell. As you can see, this on the left hand side, this is a schematic of a T cells where three major channels are shown. The calcium release activated crack channel, then voltage gated potassium channel called KV 1.3, and then there is also a important channel called calcium activated potassium channel KCA 3.1. And these channels are involved in cytotoxicity and proliferation of T cells. On the right hand side, these channels are shown to be working in a migration situation where a T cell is migrating from its reservoir to the peripheral blood circulation. And you can see the cell has changed its shape. It's becoming a polarized cell, and potassium channels seems to be present at the leading edge and calcium channels at the lagging edge or europod. So these channels involved in T cell, and there is also this evidence, the patch clamp, where I got the work on this project. We have characterized effects of monoclonal antibodies. So I wanted to bring this example in front of you to showcase that not only small molecules or endogenous agonist and derived agonist antagonist are used for this technique is used to characterize for, but also we can characterize the effect of monoclonal antibodies using patch clamp. So in these settings, we have obtained the T cell samples from the peripheral blood of head and neck cancer patients, where we were studying the effect of immunotherapy, the anti-PD-1 monoclonal antibody called pembrolizumab, and we have identified the effect of this monoclonal antibody in T cells. And you can see the example, the potassium channel currents were recorded from T cells. The black line is control, and the alpha PD1 means anti PD1. The PD1 is the targeting this therapy, and you can see the therapy was shown to increase the activity of these channels. The conductance and overall peak current was increased. So same for these two channels, the calcium activated potassium channel, the the treatment with monoclonal antibody pembrolizumab increased the channel activity in presence of treatment compared to control. For KCA 3.1 channel, same thing for voltage gated potassium channel. So this example I brought you to showcase that patch clamp is also used for screening of monoclonal antibodies. And now we have discussed a lot in this presentation about the different configuration of traditional patch clamp assay, the manual patch clamp. Um, in today's date, there is a significant automation being done in this technology. As you can see, I have brought you this slide which shows the automated version of patch clamp technique. And there are sophisticated instrument which has some robotic features. Some of them are semi-automatic, some of them are fully automated. So one example on the left hand side is shown as cytopatch. So this instrument, which looks like a typical HPLC instrument, and this has the feature to handle up to 4 cells at a time you can record. So this has a throughput of semi automatic throughput. So this is quite popular in academic institute and some industry institute as well. And there is also an instrument called synchropatch 384, which looks like a giant robot, which is actually able to patch 384 cells at a time. So very high throughput available using this technology. This is complex but also very pricey, expensive side, and that's why this high throughput technique is mostly used in industrial settings, where if you have a library of compound and you want to find the lead compounds that are active for this particular target, then this 384 assay is quite used. um, I hope this image gives you some idea about what is available there in terms of automation. And going forward, this is an example from my side, where I was involved in this project for identifying lead compounds for from a modified spartine derivative, which acting as a sodium channel blocker. I did this work in collaboration with a company called Cytocentrics. And going back a little here, so Cytocentrics used to manufacture this little device. It's called Cytopatch, which was a semi high throughput, and we identified the potential long acting sodium channel blocker in this project. So this was example. So this was an example to showcase that how automated technology was used to discover drugs and screen drugs in high throughput or semi high throughput format. And now let's take a look at the regulatory aspects, where in the new drug development the patch clamp assay is involved, and at what stage it is used. So I also wanted to shed some light on this scenario. So basically, when the drug development is in progress, especially at the stage of investigational new drug application, because we have seen that in early stage when the drug is discovered, the manual technique is very crucial to see the mechanism of the drug. Sometimes the high throughput screening approach is used to identify the lead compound, but once the lead compound is identified and you wanted to file an investigational new drug application, then also the patch clamp assay is critical, especially for the safety pharmacology, because we wanted to make sure the drug is effective but as well as safe, especially for cardiac safety. And the new drug must be tested on cardiac ion channels, especially the HERG channel, which is a potassium channel in cardiac cells. And this assay is used in vitro patch clamp electrophysiology. So regulatory agencies like US FDA requires the HERG assay data for IND application. S- And this assay is done in the good laboratory practice format, the GLP format, where things are more stringent and robust. So that's why the patch clamp data is used. And under the ICH, the International Conference of Harmonization S7B guidelines for non-clinical testing of pharmaceutical as development, this assay is used. And going forward, I would like to bring one example of a case study from the USFDA where I was also involved. Um, so once this is a case study which talks about the patch clamp assay or electrophysiology application for clinically approved drug, because I was talking a lot about the application of patch clamp in drug discovery and development stages. But what happens to the drugs that are already approved, whether this technique is still useful to evaluate them at some point? So the answer is yes. So recently, in 2021, there are several drugs which are anti-epileptics, and the US FDA was concerned about these drugs because these marketed compounds or marketed drugs were having some issues, serious issues in patients taking them. Patients having some cardiac side effects like cardiac arrhythmia, atrial fibrillation, and other things, and there are some mortalities as well. So the FDA thought this is a serious issue, and this drug should be evaluated for their mechanism. So some of the examples were like lamotrigine, which was clinically marketed by GlaxoSmithKline, carbamazepine, and topiramate by some other pharmaceutical companies like Novartis and Johnson and Johnson. Carbamazepine was from Novartis and topiramate from Johnson and Johnson. So these companies were asked by FDA to show cardiac safety data using patch clamp electrophysiology method, and they were asked to show or classify these antiepileptics on the background of antiarrhythmic drugs. So in the background of Vaughan Milliam antiarrhythmic drug classification. and I'll talk you briefly about it, not going in super detail, but at least to give you an idea, like what exactly was asked by this FDA to this company. And FDA told this company that show the safety or the mechanism of these drugs using this assay. If you fail to show that, or if there are any significant findings, then the box label will be amended, or there will be warning on the label of these drugs. Or sometimes if there are some serious issues, these drugs may be facing a withdrawal or for some certain indications, things like that. So in brief, I wanted to show Glaxo went ahead and published this data, which we also followed in our labs. And this lamotrigine was tested on cardiac sodium channels, and it was classified on the background of antiarrhythmic drug. So they were asking for two things: the potency, which is shown on the top two panels of this figure. So they wanted to show how much block it can do. So the lamotrigine was compared with the other reference drugs, which were reference sodium channel blockers like mexiletine, quinidine, and flecainide at two concentration, and they were investigating how potently this drug blocks. First thing, so this data shows that, and it was not that significant block in case of lamotrigine. And the second thing was the kinetics. In the kinetics, they wanted to show once this drug blocks the sodium channel, how quickly it recovers from the block. So recovery of the from block, and the bottom figure shows the same. So you can see there are three anti-arrhythmic drugs which were used to compare. The first drug, which is mexiletine, under Vaughan-Williams classification, the mexiletine is known for a quick recovery from the block. So the red line, which shows a very fast recovery, a mono exponential recovery, and then excuse me, and then quinidine, which is the intermediate recovery. So green line in the middle. So it looks like still mono exponential but slower than mexiletine. And then flecainide, third one, which is the slowest recovery in this panel, in this graph here. uh, Looks like a bi-exponential or biphasic. And then lamotrigine was compared, which is also appears to be fast. So Glaxo showed this data to FDA that they showed, and the penalty is just the zoomed insight from this figure C. So FDA, the GSK showed this to FDA, showing that okay, this drug doesn't appear to be a slow blocker, and it should be treated as safe. And they also supported some clinical observation from by recording ECGs from healthy volunteers, and indicating that there is no prolongation of QT interval or any arrhythmias are induced. So this way they came up with their defense strategy, and showed that their drug is safe and effective in cells and also in patients. So I wanted to give you this, and this whole data was recorded using manual patch clamp. So this example was brought in front of you just to showcase that the patch clamp assay and this electrophysiology technique has numerous application in the drug development process, the drug discovery process, and also in the regulatory process. So in pre-market and post-market situation. So with this slide, I hope the presentation gives you an idea about the technical aspects of the assay and applications and regulatory perspective. So to summarize, I have this key takeaway messages for you. So patch clamp electrophysiology is a gold standard technique to test ion channel function in pharmacology. Patch clamp assays are of critical importance in drug discovery and cardiac safety pharmacology. Also in immunology, as you saw, the T lymphocyte example. Patch clamp electrophysiology methods are available in manual and automated format and can be used based on the requirements. As you saw, but depending on the throughput requirement, you can choose either a manual patch clamp or automated patch clamp. So The last but not least is patch clamp assays are part of regulatory submissions for new drug application, IND to US FDA, and post-market safety of clinical use drugs. So I hope this presentation brings clarity about the patch clamp assay and electrophysiology techniques. And with this last slide, I would like to acknowledge my mentors and colleagues from various institutions, including my graduate school, Medical University of Vienna, Austria, my supervisor, Dr. Todd, and other colleagues at Cytocentrics for availing me the opportunity to internship in the University of Cincinnati, where I did my postdoc under the guidance of Dr. Conforti. And thank you for your kind attention. And with this note, I'm available for questions. Any?
Yes, sir. Thank you.
Thank
Vaibhav,
you.
sir,
Yeah,
there are two questions from the participants.
yeah, please.
Which drug has the highest risk of EPS?
So, can you see me now as a script?
Yes, yes,
Okay.
yes.
Is it so? The drug that has the highest risk of EPS, you means the arrhythmias, right?
Yes.
Yeah, so there are some drugs which were in the clinics, but then they were withdrawn by the FDA because of the potential and arrhythmic effects. So they increase the QT wave in the ECG, and the example is like cisapride. Once it was an antihistaminic drug, cisapride, then terfenadine is also one of the example that were once clinically approved, but then later because of their electrical activity on cardiac channels, they were withdrawn. So these are some, and then now people use these drugs to compare the investigational drug because they became a reference standard now.
So, second question from
Yeah,
another participant,
yeah.
Dipali Bhandari.
Hmm.
What drugs are used to manage EPS?
So EPS means the electrophysiology system. What
Yes,
exactly?
yes. Yes, yes, yes. I think, yeah.
So to manage means to test. That's what they are asking probably. So I believe she's trying to ask what drugs are used to manage the EPS, like which drugs we test. So if because this is we test different types of drugs in electrophysiology, but if she's particularly interested in asking like what drug is used for arrhythmia management, then it would be sodium channel blockers and potassium channel blockers like lidocaine.
Okay, sir.
Yeah.
Okay, thank you, Dr. Vaibhav Kumar, sir,
Mhm.
for your enlightening session on electrophysiological methods for drug screening. Sir highlighted the different traditional methods and the advanced cytopatch technique, clamp technique used for drug screening. Sir also covered many technical aspects of drug screening and some regulatory aspects. In that, Sir explained about how these electrophysiological methods are important with respect to the investigational new drug application, then new drug application, as well as for non-clinical testing. Okay, how as per this ICH 57B guidelines. Okay, how this importance of the electrophysiological methods for drug screening. Sir also share one case study of post-marketing vigilance of anti-epileptic drugs in 2021. Sir also associated with that particular work of lamotrigine, then carbamazepine and topiramate, and also explain about the patch clamp assay and its significance in the drug screening process. Thank you so much, sir, for your informative session to all the participants. Now, I request you to kindly accept our certificate of appreciation for from your busy schedule that you have taken time to share your expertise of this electrophysiological methods of drug screening to all the participants. Thank you very much, Dr. Vaibhavkumar Gavali, sir. And once again, thank you for accepting our invitation on behalf of our Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, for sparing your valuable time. Thank you so much, sir.
Thank you so much. And with this, I would like to thanks again to organizers, management of MS Gosabi College of Pharmacy, and colleagues for availing this platform and introducing me to the academic staff and students. I will be available for any follow-up questions via organizers, and looking forward to connect with you all, and wish you all best wishes for conclusion of this rest of the workshop. Thank you.
Yeah, sure, sir, sure. Thank you. Thank you very much.
Okay.